Então, boa tarde a todos, muito bem-vindos à nossa 14ª semana do curso. Nós estamos quase lá, né? Quase completando as 15 semanas. E eu vou passar, então, para fazer a apresentação do professor Peter e o professor Marcel, que vão estar conosco hoje. So, yet again, good afternoon, everyone. It's a great pleasure to introduce this lecture. We'll have Peter Diggle with us again in the first part of the lecture, and in the second part, we'll have Marcel Toledo from the Universidade Federal de Juiz de Fora. Peter will talk about spatial statistics and neglected tropical diseases, and Marcel will talk about impact assessment of public policies in the context of complex samples. I'd like to make a point before we start, because I would say that this course has changed from statistical methods and techniques to a kind of craft work, in the sense that real science demands from statistics lots of methods and techniques. And this is a very healthy thing, because lots of statistical methods were developed because of these demands. So in this sense, statistics serves science, providing insights or ways to deal with data on reasonable ways. But at the same time, science enriched the statistical world, provoking developments of methods, algorithms, in the very core of statistics. And I guess that today we'll have a beautiful, two beautiful examples when we go well beyond well-established methods and techniques, but showing how they are combined with real science to answer difficult, not trivial questions in the real world. So both speakers will have experience on that, and especially our first speaker, Peter Diggle. He is a real master in combining statistical principles and challenging problems in science. Throughout his career, he has published lots of papers which are real in the interspace of sciences. So in this sense, it's a great pleasure to welcome Peter again. Spatial statistics is an area of specialty of him. He's well known in the statistical community for his contributions to the field. So it will be very nice to hear him again this afternoon. So before I hand over to Peter, we made a very quick questionnaire like last time. And in the first question, I asked them whether they came across with spatial statistical before, and nearly 20% said yes to this first question. The second question is whether they see some spatial data, or they pay attention that some data would be spatial on their research areas, even if using just simple visualizations, and then this percentage goes up to 40%. Then we asked whether they know about any methods related to spatial statistics, and 13% of them said yes to this. And finally, about a fourth of them, 23%, said that they have data which is geographically referenced. So it seems like yet again we had a hot topic for today's lecture that will be close to the interests of several people in this audience. So with this, I will hand over to Peter. Peter, thank you so much to be with us again.
Thank you, Paolo. I'm hoping that, so it's quite a long title, and I'll spend the first few minutes explaining the title. And what I'll leave you with as well, Paolo has a copy of these slides, is a set of references. These are very precisely the references that I'm going to explicitly mention during the lecture. There are many other ways to explore the spatial statistics literature, but I would like to draw your attention with particular pleasure to this one here, which is the book that I wrote with Paulo Ribeiro when he was my colleague in Lancaster, and it came out of his PhD project, which was a very excellent project, and I'm pretty sure it's the first book-length treatment of model-based geostatistics, which is the methodology that I'll be talking about today. So it really is a very particular pleasure to be able to give this lecture in Paulo's university in Brazil. Spatial data come in different formats, and I've shown in this slide the three most common formats. And on the left, a lot of people would call this point process data because the data are simply a set of points. So here you have a spatial region, which is actually one of the English counties, and each circle on the map is the location of somebody who has a particular illness on a particular 5-day period. And so the data are just the points on that map, and so it's called very often a point process data set. This one in the middle is what most people would regard as a geostatistical data set. And the point about this data set is that nature didn't determine these locations. Somebody went out and deliberately went to that location there and collected some data. What they actually collected was a sample of people who they tested for a particular disease, and the colour and size of the circle is indicating the proportion of the people that they tested who were positively infected with the disease in question. So here, nature told us where the cases of disease were, and we recorded them. Here we went out to look in particular places for disease and recorded what we found. Okay, but we could have gone to anywhere else in that map. And then finally, this is a data format that you see a lot of in developed countries where people are concerned with maintaining disease registries. What that means is that the country is, in this case Scotland, is divided into a number of areas which are administrative areas. They have no particular biological significance. They're simply local government administrative areas. Whoopsie daisy. And within each of those areas, you maintain a complete record of people who are diagnosed as having, in this case, a particular kind of cancer. Now, the reason I wanted to show you this slide is partly to emphasize that the only part of spatial statistics that I'm talking about in this lecture is data of this format. But secondly, to invite you to think about the fact that these examples that I've chosen may be different data formats, but they're all concerned with the same scientific problem. Namely, what they are intended to help you investigate is the variation in a spatially continuous process over the spatially continuous geographical region in which the data sit. So here, the underlying spatially continuous surface is the risk of catching the particular disease. And it so happens that these particular people have caught it, but there are people elsewhere in the map who could have caught it. Here, the particular disease is river blindness, and because we cannot completely record all cases of river blindness, we have to go out and conduct surveys in the field, and at the locations we've chosen to survey, we can count the number of people in our samples who've got the disease and the number who have not, and that gives us an estimate again of the risk, or as it's more properly called, the prevalence of this disease. Here, we're in the fortunate position of having a complete record of all cancer cases, and somebody somewhere knows exactly where those cancer cases live, but for reasons concerned with concerns about public and private data being very sensitive, it is never revealed publicly exactly where the cancer cases are. Researchers have to work with these spatially aggregated data in a partition of the study region. But that's really just to give you a flavour of the fact that I could have spent the lecture talking about data of this kind or this kind. I've chosen to look at this data format in the middle. So let me move on and just introduce a little bit of notation for data of that format. We use the letter Y to denote measurements that we take, and they're random variables, so it's an uppercase Y, and they're at locations X, and that's a lowercase x because we're not trying to model the locations. We're saying, given the locations, what do we measure? And you can see that here we measure predominantly large values in a U-shaped region here and smaller values up here. We're interested in, however, something that we can't directly measure. It's an unobserved spatially continuous phenomenon. And I'm giving it a letter capital S, capital because I'm going to model it as a random or stochastic process. And again, it's indexed by X because it's a spatial process. So at every location X in this square, there is a value of S of X, but I can't directly observe it. I can only observe measurements Y, and those measurements Y might actually be equal to S of Xi, or they might be just approximations to it. And a very good example will be the example of measuring prevalence, which I'll get onto very shortly and spend most of the lecture talking about. But as in the previous slide, the fundamental objective, the fundamental scientific problem we're addressing is how can we use these spatially discrete finite data to say something about that underlying spatially continuous phenomenon, S of X. And that's pretty much a definition of geostatistics as you will find it in most textbooks. Geostatistical problems could be concerned with design. How should I have chosen these locations? If I was free to choose these locations anywhere in the square, why did I choose those particular ones? How could I choose them in order to make the answers to the question of interest, which is what I can learn about S of X, as precise as possible? We could have a problem of estimation, how to investigate relationships between what we measure and where we measure it, and whether it relates to covariates that we might measure. For example, does disease prevalence vary with height above sea level? Does it vary with the socioeconomic development of the community in which you're measuring disease? And this would require you to investigate parameters, to estimate parameters that describe regression relationships. And finally, and most importantly for this lecture, the problem that we're addressing is very often in geostatistics a prediction problem. We're not usually very interested in parameters in the classical statistical sense. We're usually interested in trying to find out, as I've indicated in the previous slide, about an underlying stochastic process that we can't observe directly. And that is a prediction problem, not an estimation problem. And as I shall show you, the distinction between estimation and prediction turns out to be very important. A little bit of history. Geostatistics has its origins in the mining industry in South Africa, and its earliest exponent was a professor in the mining engineering department of the University of the Witwatersrand, Professor Dani Krieger, and he promoted the value of using statistical methods to answer questions of the kind that my geostatistical example exemplifies, namely, if this square here on this map is an option to buy a piece of land and explore it for mineral wealth, if you are allowed to take some measurements before deciding how much to pay for that piece of land, and these were your measurements, and this was the quality of the ore that you could extract from those measurements, this might be gold, for example, then first of all, if you could actually buy perhaps just half the square, you'd probably be more interested. And secondly, if there's no option but to buy the whole square, you might want to focus your mining effort in this area here rather than this area here. And of course, you could look at whether as a whole, on the basis of these data, you would predict that the return that you get out of the mine is going to be more than the price you pay for it. Geostatistics made its way from South Africa to France, where Professor Georges Matterhorn led a statistics department, although he wouldn't have called it that, but certainly a department doing statistical work of a very advanced and ingenious kind in the French National School of Mines. Again, the connection is with mining, and the motivation is to be able to predict the final economic yield of a mine based on some exploratory samples taken from the area where you're proposing to dig that mine. And Matterhorn was a very fine mathematician and erected a very detailed and complete theory of geostatistics, which essentially was self-contained within the French intellectual school and made little or no connection to mainstream statistics in general. About the same time in another part of Europe, in Sweden, Bertil Matern here was a professor of statistics in the Royal College of Forestry in Stockholm. And so now the interest is not so much in mineral exploration, it's actually in forestry exploration, because forestry was a major and still is a major part of the Swedish economy. And the same kind of questions arise in predicting how, firstly, to maximize the yield of a forest, how to plant it, and as the forest grows, how to measure and acquire data that can give you a prediction of the eventual value of the forest. Again, Bertil Matern worked in a college of forestry and published essentially nothing in the mainstream statistical literature, but his work was of seminal importance, and as with Matern's work, it was picked up by statisticians and brought into the statistical mainstream in the following 2 decades. Jeff Watson wrote some papers in the geological literature pointing out that geostatistical methods were really stochastic process prediction methods. Brian Ripley, a British statistician, wrote one of the first books on spatial statistics in 1981, in which he devoted a chapter to a very condensed account of stochastic process prediction, which was geostatistics except by name. And Noel Cressy, in his very large and very well used book on spatial statistics, including but not restricted to geostatistics, classified geostatistics as one of the 3 main branches of statistical investigation, the others being point processes and what he called lattice processes and what I would call discrete spatial variation. And not coincidentally, those 3 areas correspond to the 3 pictures I showed earlier: point processes, geostatistics, and, in Cressy's words, lattice data, in my words, discrete spatial variation. So there we have the background to spatial statistics in general, what geostatistics is within that general area of spatial statistics, and a little bit of history of how it came to be developed in different places at different times by different people until it is now a recognized mainstream statistical topic. And at this point, myself and 2 Lancaster colleagues, Rana Moeed and Jonathan Thorne, wrote a paper in which we introduced the term model-based geostatistics to mean nothing more than the application of general principles of statistical modeling and inference to geostatistical problems. Okay, so we have statistical principles that we can apply to any scientific method. And the goal of model-based geostatistics is to apply those principles to geostatistical problems. So let me talk a little bit about this slide. Firstly, on the right-hand side, to remind you of the notation, S is our unobserved, spatially continuous process. We can't directly measure it. It will represent throughout the remainder of my lecture the prevalence, which implies the risk, of a particular disease at any arbitrary location in a geographical area of interest. We would like to know about S, but because we can't directly measure it, we need to go and collect some data that's relevant to it. The data Y don't have to be exactly values of S, but they have to be related to S in order to enable them to be useful to predict what we would be able to see if we could indeed directly measure S. So Y are our information, which we're going to use to find out about S. And we'll do this in a hierarchical manner as follows. We will say we have a stochastic process S, which is of scientific interest, and we have a stochastic process, or at least data set Y, which allows us to find out something about S. And so we need a statistical distribution, these square brackets mean the distribution of, 2 stochastic objects, S and Y. And we know from elementary probability theory that we can factorize a bivariate distribution in 2 mathematically equivalent ways, namely the marginal of S times the conditional of Y, or the marginal of Y times the conditional of S. And before I proceed through the rest of that equation, I now want to take you to the triangle on the left-hand side of this slide. This triangle is meant to indicate the role of statistics in scientific method. At the apex of the triangle, we have the state of nature. Nature is what we wish to understand. And scientists progress their understanding of nature in two quite different ways. They can devise theories of science, and in so doing, they will produce a model of a process which represents a mathematical abstraction of how they think nature is behaving. Scientists can also collect data, but they run experiments or observational studies resulting in data, and they can build models for their data. These are statistical models which say what probability distributions will accurately describe the scientist's data. Statistical inference is the process of using the data to find something out about the underlying process. Now, the way we do that is as follows. It is certainly true that the process exists without the data. Disease prevalence exists. Gold under the ground exists. It is not true that the data exists without the process. You cannot collect data on prevalence unless there is a disease. You cannot collect data on gold unless there is such a thing as gold. In other words, it makes sense scientifically to factorize this bivariate distribution, coming back to the right-hand side of the slide, as a marginal of S, because that does exist in its own right, and a conditional of Y given S, because that's how we collect the data. Given the state of nature, S, that will determine the properties of our data. Okay, now that's the right way to build a model, in my opinion, and it's a hierarchical statistical model in which it is trying to combine a scientific representation of the process with a statistical representation of the data conditional on the process. But of course, the inference that we want to make is the other way around, because we want to find out about S, right? And in order to find about S, we want to know about the distribution of S conditional on Y. But we can do that because we have something called Bayes' theorem. And Bayes' theorem means that if we have the distribution factorized this way round, we can equally well invert the conditioning and get a conditional distribution for S given Y, and that becomes a conditional distribution of T given Y. Now, I apologize for missing an intermediate step in the notation, but T is there because it's meant to be the target of your inference. And the target for your inference will be something to do with the state of nature, but it won't necessarily be the whole of the state of nature. It may be some particular property of the state of nature. So I've used a different letter T, but that's a slightly opaque transition of notation, for which I apologize. Now, there's nothing in this slide here that includes parameters, so it's not explicitly about parameter estimation, but in practice, all of the models that we will come up with will have parameters that we need to estimate. And the way that I and my colleagues choose to estimate our parameters in most applications is through Monte Carlo maximum likelihood. Monte Carlo because the likelihoods that we encounter in this area are typically analytically intractable. If your preference is for Bayesian inference, then you can put Bayesian priors on your model parameters, and again using Monte Carlo methods in practice, you can obtain posterior distributions for your parameters by combining your prior distribution with the likelihood. The point I would like you to agree with me on is that the likelihood is central to the inference. Whether you implement it through maximizing it or integrating it, the likelihood is the core of what's going on. All right, and how you use the likelihood is a matter to some extent of personal taste. Okay, so that's what we're going to be doing for the rest of the lecture. So let's make this a bit more tangible. S is the state of nature. Y is our data. T is some property of S, which is our target for prediction. For scientific reasons, the natural way to parameterize the model is as the unconditional of S times the conditional of Y given S. For prediction, we need to invert the conditioning, and having inverted the conditioning, we acquire a distribution for our target, conditional on Y. That's the predictive distribution, the distribution conditional on the data. And to deal with parameter uncertainty, we can either plug in an estimate of theta, or we can weight different values of theta according to their sampling distribution, if we believe in maximum likelihood, or the posterior distribution, if we believe in Bayesian inference. And operationally, we will usually, in my experience, for the problems I'm talking about tonight, get very similar answers. Okay, so here we have the African country of Liberia. And you've seen a picture like this before in my earlier slides, I won't keep embarrassing Paolo by mentioning his name, but this picture is produced by a very nice R function that Paolo wrote many years ago. And it indicates, again, the variation in the prevalence of a particular disease. The disease here is river blindness, which is a serious public health problem throughout tropical Africa in poor rural communities. And you can see that river blindness has spatial structure across the country of Liberia, because red and large discs are areas of high prevalence, browns and yellows are intermediate prevalence, greens and especially blues are low prevalence. So without doing any statistical analysis, you can see that river blindness is very uncommon near the coast, but as you move away from the coast, it generally gets worse. But it doesn't get worse in a way that you could easily characterize by a simple regression equation. There are spatial patches of high intensity, and there are spatial patches of low intensity, which are the same distance from the coast. And also there is variation within these red patches. There's a little yellow dot creeping in amongst the red dots. So you can see that there's a lot of stochasticity in this map, but equally there is spatial structure that you would like to extract from the data, and you probably can't do it with a simple regression equation. So you need this notion of a stochastic process that describes the underlying true risk of disease, and stochastic data, the circles, which give you partial information about that underlying continuous process. And the three statistical questions that immediately arise is, if you are being advised on how to conduct this study in advance, how many samples would you have advised these people to take? Where would you have advised them to put them? And how would you analyze the resulting data? And that's what we'll now proceed to do. It's very important to recognize the low resource settings element of my title, because when you approach questions like how many samples should you take and where should you take them, you might make a suggestion that, for example, you should space your samples evenly out across the country, right? I think many people would think intuitively, if you want to understand spatial variation and you've got, let's say, the opportunity to take 100 samples, you wouldn't have them irregularly distributed like this. You'd put them on something like a regular lattice. Well, tell that to the man on the motorbike, who is a field epidemiologist, who is on a 4-week trip away from home, navigating his way through countryside like this, trying to collect data to help fight river blindness in Liberia and other African countries. You have to take a very pragmatic view of experimental design when you're working in low resource settings. It's still valuable to say what the theoretically optimal design might be, but it's much more useful in practice to then recognize what the practical constraints are in implementation of field work, and to accept that often you will be faced with data that are generated by a suboptimal design, and then, as statisticians always do, you will turn your attention to analyzing those data in the most efficient way possible. Having said that, we will be able to advise the man on the motorbike to spend less effort next time he goes into the field to try to collect data. Schematically, this is what's going on in a geostatistical model. If you don't like algebra, this is your definition of the geostatistical model that I'll be using in the remainder of the lecture. Namely, if this is a transect running from west to east, and you are moving along this transect, you will perceive that it's getting hotter and wetter. And because we're going to be dealing with vector-borne diseases spread by biting insects that like things to be hot and wet, you would predict on the basis of the environmental gradient, which you can think of as a covariate, that you would be encountering higher risk and therefore higher prevalence of disease. Every so often, you encounter a village, and when you do that, you collect data. And you record in this village about a 35% prevalence from a sample of, let's say, 100. In this village, more like 30%. Here, 50-something%. Here, 60-something%. But because you know your sample size and you know the theory of the binomial distribution, you can work out the binomial sampling error associated with your data. And if that sampling error is compatible with the departure of your observed data points from your best regression model, you've got a good statistical model, and you can stop listening to the rest of this lecture because you can use classical regression modeling. If, however, the sampling errors appear to be larger in a stochastic sense than are compatible with binomial variation, and they appear to show spatial structure, then what you have in this dashed line here is the unobserved stochastic spatial process, which is what we can actually make inferences about using geostatistical methods. So the message from this slide is actually that when you want to do a geostatistical analysis, the first thing you should do is forget about geostatistics and do a regression analysis. And then, if your residuals pass all of the usual checks, including crucially that there's no correlation amongst the residuals, you're done. If, as in my experience is commonly the case, they do not pass those tests, and in particular the data do show spatially correlated residual variation, you need geostatistical methods to answer the questions. For the statisticians in the audience, this slide is identical to the previous slide, but expressed in algebraic notation, which will either be more familiar to you or completely abhorrent to you, depending on your scientific background. So we have all of the ingredients that I hope you're now beginning to be familiar with. We have a latent spatial process S of x, latent meaning we can't observe it. We actually allow as a separate latent effect a set of independent quantities which don't vary spatially in a correlated manner. Because remember I said that if these things are not spatially correlated, you shouldn't force it upon them. I didn't say that, but I certainly meant to. We allow for them to be spatially correlated, but if they aren't, we're happy to put our efforts into this U of x term and put less focus or even ignore the S of x term. But between these two terms, we're accounting for spatially correlated and spatially independent variation that we cannot explain by a regression model. And what is our regression model? Well, because we're dealing with prevalence data, we're dealing with binomial sampling, so we're actually dealing with a generalized linear model, and the classic example of that for binomial data is a logistic regression model. And the geostatistical extension simply has these random effects, which are associated with locations x, added to the regressors d of x, which are the hotness and the wetness, the environmental variables, and the regression parameters beta. And if we apply that paradigm to the Liberia river blindness map, this is what we get. And you can see, and qualitatively there are no surprises here, that prevalence was low, 10% or less near the coast, increasing to high, more like 30% in areas further away from the coast, but not uniformly so. Here are some areas quite close to the coast where prevalence is quite high, and so on. Now, the first question any statistician ought to ask of this map is how precise is that map? And I have over the years come to the conclusion that quoting standard errors and variances on a map like that is not very useful. A more useful thing is to map something that might have some relevance to public health policy. And in the case of river blindness, there is an internationally recognized policy promoted by the World Health Organization that areas where the river blindness prevalence is greater than 20% should be prioritized for intervention. Remember the subtitle low resource settings. It would be wonderful to make treatment for river blindness available to everybody in Liberia. It is not possible economically or logistically, at least certainly not except over a very long time period. And so you have to prioritize the high risk areas. So given WHO's policy, to me, a more useful way of conveying uncertainty in this map is to say, rather than just saying these areas are above 20% prevalence and these areas are below 20%, how confident are you? How likely is it that each point on the map is above 20%? And here you have on the right-hand panel here the answer to that question. And this matters because the data points are unequally spaced and the number of people in each sample point are different. And so the precision varies in ways that are hard to convey from a simple binomial formula. And this is, to me, a better way to convey them. Areas where we've got light pink, we're saying we may not know precisely what prevalence is, but we are essentially certain it's less than 10%, 20%, I beg your pardon. Areas that are dark green, again, we may not know exactly what the prevalence is, but we're pretty much certain it's above 20%. And apart from actually telling you the answer to the policy relevant question, this also tells you that if you wanted to demarcate the whole of Liberia as being high priority, i.e. greater than 20%, or low priority, i.e. less than 20%, the map also tells you where you need to collect more data, because you need to collect more data in the zones of uncertainty. Right? Now, if the WHO's policy had been to regard areas with prevalence greater than 10% as of high priority, then the zone of uncertainty is much smaller, because we're pretty certain that a very, very large fraction of Liberia has a prevalence greater than 10%. So I would recommend, if you're trying to convey uncertainty in a geospatial map, to produce these so-called probability of exceedance maps at a range of thresholds that are relevant to the practical question you're trying to answer. Okay, now, what are the advantages of using model-based geostatistics for problems of this kind? Well, the most obvious one is that in low resource settings, the kind of registry data that I illustrated earlier with data on Scottish lip cancers do not exist in low and middle income countries, and they certainly do not exist in most tropical African countries where I tend to do most of my work on these diseases. And we can build into our analysis some modeling assumptions to compensate for the sparseness of the data. We can talk to entomologists about what environmental conditions would promote successful breeding of the vector that spreads the disease. We can talk to social scientists about how impoverishment is likely to affect how people are exposed to the disease. And so we can build in these modeling assumptions by a dialogue with scientists, basically implementing the triangle. That scientists tell us what they think is driving the process. We then collect data, and as statisticians, we analyze those data as efficiently as we can in order to answer the scientists' questions. And because resources are always overstretched, statistical efficiency is paramount. And we know, at least since 1960, that classical survey sampling methods are generally very inefficient when data are spatially correlated, but they are still the WHO standard for assessing neglected tropical disease prevalence in Africa. And in my humble opinion, they are not fit for purpose. Why not? Because with geospatial analyses, we borrow strength in the sense that if I collect data at a particular location, that doesn't just tell me about that location, it tells me about nearby locations. And that's why I can attach a color, not just to the sample data points, but to the nearby data points. Because what I'm implicitly doing is I'm invoking what's sometimes called the first law of geography, which is that everything is dependent on everything else, but close things are more strongly dependent than distant things. And as a result, we get a much greater efficiency than if we rely on classical survey sampling methods. I'll give you an example of that in terms of this simulated example. This is a prevalence surface that I've simulated, and the area-wide prevalence, if you integrate that surface over the whole square, is 41.7%. And if you measure prevalence at some sampling locations, you can apply classical survey sampling methods and come up with an estimate which is pretty good, except that your uncertainty, your honest uncertainty, is quoted using the textbook methods as plus or minus 8.6. And if you do a geostatistical analysis of exactly the same data, you get almost the same estimate, which is typical, but a much smaller standard area. And what I'm going to show you in the remainder of the lecture is that we get very similar improvements in performance in real tropical disease settings, where the correlation is substantial, and therefore, and you can see it very clearly on this picture, that single data point is telling you about the value in this whole area around it, because the underlying surface is spatially smooth. So this extra efficiency comes from two different things, which is why I said earlier it's important to understand the difference between estimation and prediction. The obvious gain in efficiency is because we're exploiting spatial correlation, which is that data at one location are informative of prevalence at neighboring locations. And by fitting a model, rather than just picking an arbitrary smoothing algorithm, we allow the data to choose the optimal definition of neighboring. The extent to which data at one location can predict results at other locations is something we estimate from the data. We don't impose it on the data. But the other thing is that you have to ask the right question. And the classical survey sampling approach is an estimation approach. It's actually answering the question, how precisely can we estimate prevalence under repeated realizations? The geostatistical approach is saying, how precisely can we predict prevalence in this particular realization? So it conditions on the data. There's no variation in the data in the geostatistical analysis. It's conditioned on, and it predicts what we would have seen if we'd been able to interrogate the process directly. Whereas the classical approach is saying, I can estimate for you how variable your estimates would be if you did it again and again and again, but I'm not going to do it again. I've done it once and I want a condition on my knowledge. And that's why the predictive variance is much smaller than the estimation variance. Okay, so on to neglected tropical diseases. And this slide, which I don't wish to spend time on, is simply there so that those of you who do want to follow up on the topic in general, which is one that is a very rich source of satisfying problems for statisticians to work on for the improvement of the health of poor populations worldwide. And you can see that neglected tropical diseases exist primarily in the tropics, as you would expect. And although Brazil looks bad here because it's simply a very large geographical footprint, the worst problems by far for communities suffering from neglected tropical diseases are in Africa, with pockets, well, sometimes quite large pockets, but certainly areas in South Asia and Latin America that are also substantial public health problems, but certainly the majority of the really catastrophic consequences of neglected tropical diseases are in the African continent. When you work with neglected tropical disease, you have to understand that words which have a very similar meaning in the English language have distinct and really quite different meanings in Sustainable Development Goals as expressed by the World Health Organization. Eradication means the permanent reduction of worldwide incidence of a disease to zero. And to the best of my knowledge, the only disease that's been permanently eradicated is smallpox. Elimination means reduction of incidence in a specific geographical area to zero. In other words, there's no permanence and there's no worldwide. So you can certainly get rid of a disease in a particular area at a particular time by giving everybody protective medication or curative medication, but you cannot be sure that the disease doesn't still exist somewhere else, and you cannot be sure it will come back if you stop treating people. And then finally, and this is what I work on, elimination as a public health problem means reducing the incidence in a specified geographical area to an agreed level. So it's a pragmatic or realistic view of the world that if we can get disease down to a low level of prevalence, then we can certainly more effectively treat the much smaller number of people who need it, although we can't prevent people getting the disease altogether. And we have to sort of settle for that realistic goal. And that's what the WHO, again, has done in terms of each of the neglected tropical diseases. It sets policy guidelines as to what it would regard as a successful intervention to eliminate a particular disease in a particular country as a public health problem. And the one I'm going to take you through is lymphatic filariasis in Ghana. Lymphatic filariasis is a disfiguring disease. It doesn't kill, but it very seriously affects quality of life, and it affects many, many millions of people in tropical, poor, rural areas of Africa. So this is Ghana, one of the countries of West Africa. Ghana is divided into districts, and where I'm going to analyze some data that were collected on the prevalence of LF in each of the Ghanaian districts, or at least in most of them, before any interventions were carried out. I'm then going to simulate what the geographical distribution of that disease ought to be if and when the intervention has been rolled out sufficiently to get the country to a point where the WHO would accept that it has achieved elimination as a public health problem. And that's a classic example of a statistical design problem in which you have to make an assumption about what the true state of nature is, and on the basis of that assumption, you design a study that will be able, with sufficient precision, to tell you whether your assumption was right or wrong. So we need to be able to say, this country believes it's now just about at the point where it has eliminated lymphatic filariasis as a public health problem. How can we design a study that would give them sufficient data to answer that question with reasonable precision and be affordable? So obviously, if money was no object, it's an easy problem, but money always is an object, particularly in low resource settings. And I'm going to then compare two design strategies. I'm going to compare what we would get as our answer to the question using current WHO guidelines, and I'm going to answer the same question, but using model-based geostatistics. And so the question is going to be, for each of these 216 districts in Ghana, has that district successfully achieved elimination of lymphatic filariasis as a public health problem? So firstly, what are the current WHO guidelines? Well, for each of the 216 districts, you know the population of the district, total population. So you go to some tables provided by the WHO, and they will tell you how many villages to sample, how many children to test in each village, and they will give you a magic number. And they will say you simply add up the results of all the tests from all the children in all the villages, and if the number of positive test results that you get is greater than 23 or 27 or whatever the number the table tells you, you will declare that you've eliminated the disease. If it's bigger than that number, you will declare that you have not indicated. You will declare that you have not eliminated the disease. So elimination is not indicated. So it's a very deterministic process. All it tells you is how many villages to sample, how many children per village, do some statistical arithmetic on the test results, and give a simple yes/no answer. Now, model-based geostatistics looks like harder work, but it really isn't, because all the hard work, if you remember the man on the motorbike, is in actually collecting the data. Once you've collected it, analysing is, relatively speaking, a very cheap and easy operation. So what we do is we look at all of the communities throughout Ghana, okay, and we say what we're really trying to predict here, our target, is the prevalence in each community weighted by the population size of that community divided by the total population. And we do that for each district. So that is our target. That would be the proportion of people, children in that district that have lymphatic filariasis. And the WHO policy directive says that elimination as a public health problem is defined as that quantity T is less than 0.02. So basically 2% prevalence averaged over the district, that district has eliminated LF as a public health problem. And of course, we, being statisticians, are going to make a probabilistic prediction. because it's a probabilistic problem. So we will use our methods to sample from the predictive distribution of T. If you remember, I said earlier that our target is always some property of the underlying prevalence surface. Here, that's the property. P for prevalence rather than S is the prevalence that exists throughout, but in particular, it exists at every community, whether or not we've sampled that community, and we know how big each community is, and therefore, if we've got a predictive distribution for the surface P, we've also got a predictive distribution for T that we can sample from, and we will choose a probability threshold and say we will declare elimination if the probability of elimination is at least Q. So you can think of Q as a confidence level. So typically we might make it 0.9, 0.95, 0.99, and we're saying we can't be certain, but we're reasonably certain that this district has succeeded in eliminating the disease. So these are the data that we have available to conduct our analysis, to design the study and to evaluate its properties. 403 locations, so an average of about 2 per district, spread more or less throughout the country. And as you can see, before, baseline means before any intervention, then the prevalence, remembering that 2% is the threshold for elimination, the prevalence was already low in some areas, but was very, very much higher in others, particularly in the extreme north and south of the country. If we want to understand spatial correlation, we need first to estimate it from our data. And because our data come as an irregular spatial distribution, rather than nicely lined up in pairs, we can't directly calculate a sample correlation. If I have 2 samples on random variables and I want to know if they're correlated, I just line them up and I calculate the pairwise correlation. For geospatial data that's irregularly distributed, although you can't do that, what you can do is you can look at differences between values, and you can look at those differences in terms of how far apart the corresponding locations are, and you can estimate the variance of the difference because the square of the difference is an estimate of the variance, assuming that the underlying random variable has constant mean. So if this is a regression residual, which it is in practice, half the squared difference between 2 values estimates this quantity V of U, and that means that we can estimate the variance multiplied by 1 minus the correlation. And so then if we look at pairs of points which are a different distance U apart, we can see that pairs of points close apart tend to have small squared differences. The variance of the difference is small if the points are close together. Pairs of points very far apart, 200 kilometers, much bigger variance because there's no correlation, because the variance here is a factor times 1 minus the correlation. So as the correlation decays, this V of U gets bigger. And in particular, this blue region here is a 95% envelope for what we would expect to see if there's no spatial correlation. And so what you have here is confirmation that there really is spatial correlation, a pretty clear visual indication that that correlation is decaying with increasing distance separation because the variogram is increasing, but it's essentially decayed to 0 by the time we've got to 100, 150, 200 kilometers. And I'm being deliberately vague here because I don't regard this as a tool for estimation. I regard it as a piece of exploratory data analysis. As I mentioned, what may seem a long time ago now, when I want to estimate parameters, I use the likelihood. Now, some of you will have been at my longitudinal data lecture a few weeks ago, in which we also used the variogram for looking at correlation in time rather than in space, but the principle is the same. And I know Paolo has given you a preliminary short lecture in which he also introduced the idea of the variogram. But basically, this is an empirical tool for visualizing spatial correlation in order to get reasonable starting values for the parameters in a stochastic model and to confirm that there is indeed some spatial correlation worth estimating, indicated by that pattern. So we apply our methods to produce now a spatially continuous map of prevalence at baseline. And as expected, there are substantial areas which are below 2% in predicted prevalence and substantial areas that are above 2% in predicted prevalence. Everything from the pale blue onwards through to the yellow and the red is above the threshold. And we can plot that in a way that administrators are more comfortable with by aggregating two districts. And it's a small technical point, but what we don't do is aggregate the data. Actually, it's an important technical point. We don't aggregate the data to district level, we aggregate the predictions, okay? Because you cannot, well, you throw away information if you aggregate the data, whereas if you aggregate the predictions, you're preserving all the information. But as in my previous example, what I really want to know is, what am I saying about compliance or not with the elimination threshold of 2%? And so here you can say the probability of elimination, right, is essentially 0, where you see red in the map, but there are some areas where, without any intervention at all, I'm fairly confident that the disease has been eliminated. I'm at least 90% confident that the disease has been eliminated. At the district level, there are only a couple of districts where I'm 90% or more confident that the disease has been eliminated. And this matters because operationally, a district is the unit that the public health authorities will have to manage. So it's no use telling them that they don't need to do anything here, but they do need to do something down the road, because that's not operationally feasible. So that district only has between 80 and a 90% probability of having achieved the elimination threshold district-wide. So operationally, the distinction between spatially continuous and spatially discrete matters because of the practical constraints of having to continue to treat or not throughout a district. You cannot make, it's not feasible to make decisions at a village by village level. It's very difficult in practice, and it's also problematic from an ethical perspective. If you give your one village treatments but don't give a neighbouring village treatment, and people are obviously free to move between villages, you're asking for trouble. Okay, so now a little bit of algebra again, but it's actually a very simple piece of algebra, because in order to do our design calculation, we need to now simulate what we think lymphatic filariasis looks like now in Ghana after many years of treatment for the disease. And so we work on the log odds scale because we fitted a logistic regression model, and we do a very simple adjustment. We shift the log odds by an amount that results in a countrywide average prevalence of 2%, because the country believes they're just about at the level of elimination. And they believe that because they have some understanding of the state of health of the people that they're responsible for. They haven't got hard data on it, but they, as they go about their business as public health professionals, they can get a sense of lymphatic filariasis being less of a problem than it was years ago, and they have decided they would like to run an elimination study. They would like to see whether in fact they've achieved elimination countrywide or just in certain districts or not at all, where they'd certainly hoped to have achieved it in some districts because they didn't need to treat at all for that. So we project our prevalence maps, and our projection is based on preserving the spatial structure but shifting it so that the nationwide average prevalence is 2%. And so on a point map, we now see that we think very large part of the country is below 1%, never mind 2%. We know 2% is the second bluest colour, and most of the districts are in the two darkest blue colours. But there are quite a few countries where we're really not so sure, and one or two where we're actually fairly sure that it's not above. I'm sorry, I do apologise. That was the prevalence. This is the slide I should have been talking about. Okay, this is the probability that prevalence has been achieved. And you can see that now the dark areas, dark blue areas are the areas where we're at least 90% certain we've got below the 2% thresholds, and the dark red areas are where we're at least 90% certain we haven't. And if we translate those to districts, we can see, again, a fair swathe of districts where we're at least 90% certain that the district-wide prevalence is less than 2%, and therefore they've eliminated the disease, and a substantial number of districts where we're pretty sure they haven't. They've certainly reduced it, but not to 2%. Okay, sorry about that mistake. I hope we didn't throw too many people. So how do we know how well this is going to perform in practice? We have to evaluate the predictive performance by simulating the test results and applying both the WHO guidelines to those results and our model-based geostatistical methods to those results and comparing the answer. And the way we do that is we construct tables of true and false, positive and negative indications for each of the 216 villages. And we calculate the positive and negative predictive values. So the positive predictive value is the probability that you will have a correct positive indication. In other words, a district that you say has a positive disease status really is positive, and the negative predictive value says what's the probability that a disease that you state, a district that you state has negative status really does have negative status. Now, for the WHO method, you just get one answer for each district, yes, no. For our method, you get a probability distribution for each district. So to turn it into a decision to compare with the WHO method, you have to decide at what probability, threshold Q, you will declare elimination. So in your mind, you can think of Q as being a high probability, right? Mathematically, it can be any probability, but to be useful, you'd want this probability to be high, saying, I'm pretty certain this disease has been eliminated, so I'm going to declare it so. Now, because we're comparing a single number with a whole range of numbers according to what we choose for that critical threshold probability, we have to use an ROC curve to compare our methodology. And I want you to focus just on the top left panel in this slide, because we're nearly done now, you'll be glad to know. And there's quite a lot of information in this slide. Firstly, PPV, the percent correct positive indications, is on the y-axis, right? So ideally, we'd like that to be 100%, right? Now, NPV is the percent correct negative indications, and on the x-axis, we've got 1 minus NPV. So ideally, we'd like that to be 0%, right? We'd like all our predictions to be correct. So we'd really like to get a result in this top left-hand corner. Sorry, I told you to look at this one, didn't I? Right? Now, oopsie daisy. You will only ever do that if you have a prodigious amount of data. The WHO guidelines tell you how much data you should collect, and they produce that X there. So your positive predictive indication is very good. It's 95.5%, but your negative predictive value is really pretty poor at 27%. And the WHO people have presumably designed their study with this in mind, that they have put a high premium on getting a high positive predictive value, and they've sacrificed quite a lot of negative predictive value to achieve that. Now, with an ROC curve, you can choose whatever balance you like, because you can basically say, I'm basically going to just never say anything is positive. I'm just going to do all negatives, and then your negatives are all correct, but of course your positives are all wrong. So that's pretty hopeless. You could do it the other way around, declaring everything positive, and that doesn't tell you anything. What's useful is that you have a curve between here, and remembering that the ideal is to be up in this top left-hand corner, you're probably going to recommend using a threshold probability that puts you somewhere in this region where you're as close as you can get to the ideal position. But depending on the importance of positive and negative errors, you might want to skew it this way or that way. But from a statistical perspective, the thing that really is striking about this picture is that it doesn't really much matter where you are on this curve. There's a huge opportunity to do better than the WHO guidelines, right? Because anywhere in this whole sector beats the WHO on both positive and negative predictive value. Now, what gets more interesting and potentially much more important practically is that if we reduce the number of sampled locations to a factor of 1/5 the WHO recommendations, we still do better. We can still beat the WHO guidelines on both positive and negative predictive value. Why stop while you're winning? if we decrease the number of sampled locations to 1/10 the number of locations that WHO recommend, we don't do so well. We don't do much worse, but we do worse. However, if you want to argue, and I'm not particularly pushing this, but my colleagues at WHO say it's a reasonable argument, which is that if you can afford to sample fewer locations, then you can compensate by sampling more children at each location, because your resource limitation is the person's time in going out into the field. Now, if you actually make the total number of children constant, then it turns out that if you start in the same place, which is sampling the number of locations recommended by the WHO and the number of children recommended by the WHO at each location, and then when you produce 1/5 as many locations, but compensate by sampling 5 times as many children, you essentially preserve the ROC curve. And if you then go down to 1/10 the number of locations and 10 times the number of children, you again preserve the ROC curve. The reason you do in this example, and it's not a universal truth by any means, is that we do have very long-range spatial correlation. And so we really can cut down quite sharply on the number of locations and compensate by adding children at each location and really get just as much information as we started with, almost as much anyway. So those are not small savings. Those are very large savings, and imply that you can really go from 30 districts down to 3, which is a 10-fold saving of field effort, perhaps increase the sample sizes at each school a little bit, not as much as 10-fold, to get you back up to the level that WHO regard as acceptable. And this gives you an opportunity to really make a lot better use of limited field resources. So that's the end of the story for tonight. Just to conclude, I've really scratched the surface of modern model-based geostatistical methodology. As I said at the start, Paolo and I wrote a book on this topic 13 years ago, and there's been a lot done since. And the methodology that I've described tonight has been extended in many ways, including using different diagnostic tools and combining the data from those. Combining randomized and convenience survey data. You can't, for resource reasons, expect to do a classical randomized trial of interventions in this context, but if you can do a small randomized sample and combine it with opportunistically collected data, you can often do better. Because of the cost in time, more than money, of field work, there's a lot to be said for going to survey more than one disease in the same investigation. So that gets you into multivariate models. There's also a lot to be said in the age of mobile data for field workers like our friend on the motorbike, sending data in remotely to a central access point where it can be analysed, and the results of the analysis advise the field workers where to go next. That's called adaptive design. The diseases that I work on are mainly endemic diseases where time is not a factor, but in other diseases, for example, malaria, which is a very non-neglected but still important tropical disease, spatiotemporal extensions are important. And again, exploiting mobile data feeds, there are potential applications of these methods for ongoing real-time surveillance of a wide range of public health conditions in both developed and developing country settings. In terms of practical tools, the software I've been using tonight is an R package PrevMap, which my colleague Emanuela Giorgi and I wrote specifically for disease prevalence applications. I also still use the R package that Paolo wrote, GOR, which I still think has a much nicer user interface, but it doesn't have the functionality that PrevMap has. And what we're doing in Lancaster at the moment is developing more user-friendly interactive user interfaces so that people who are not experts in geostatistics can apply the methods to population health problems in country. And some of the more recent developments are in this second book, Diggle and Giorgi 2019, which is also among your references. But at that point, with apologies for going slightly over the hour, I'll be very happy to answer any questions.
Great, Peter. Well, thank you so much. It's always so nice to hear you. Peter, as I told you, a good part of our audience are colleagues from the health sciences. So I just want to tell you that I have some messages here that people are very pleased to see the problems being treated and discussed the way you did.
Thank you.
I have some questions. I start with a slightly more technical one about how flexible is the Monte Carlo likelihood you mentioned in terms of dealing with this different distributions.
Yeah, okay. I guess the answer depends on whether you're talking about under what circumstances does it work if you've got the software, and for what circumstances have you got the software. PrevMap fits essentially three classes of models, and it will not be a surprise to most of your audience to know that those are linear Gaussian models, logistic linear binomial models, and log linear Poisson models. The method is completely general and very elegant, and I think it's basically means that you can use the very powerful tools of Monte Carlo inference for solving intractable inferential problems, either using Bayesian or classical frequentist likelihood-based methods according to choice. There are no easy solutions to Monte Carlo inference. The onus is always on the user to check convergence of their algorithms, to do diagnostic checks that the algorithm has converged to a sensible point if you're doing maximization, and in my opinion, to check that all parameters and combinations of parameters are well identified if you're being Bayesian. So it's not a magic bullet, but the method is completely general. The tools in PrevMap are restricted to those three classes of geostatistical model: linear, log linear, and logistic.
Great. Peter, on a small diversion in terms of setup, a colleague is asking about analyzing data spatial data, when you have different layers, like soil layers, but not many, not a long series to become a spatial temporal analysis.
Right. So yeah, I'm sure. I would actually say that for that kind of data, that's what I call not three-dimensional data, but 2 + 1 dimensional data. And so the other way to think about that is that you can think of it as a multivariate process in which there is a spatial process at each of several layers. And if you're dealing with just a few layers, as in pedometry, then it's probably, I think it's okay to think of those as discrete horizons, you know, surface, subsoil, base rock, whatever, and to think of it as a multivariate spatial problem. But the multivariate structure may well itself be set up in a way that respects that there is actually an ordering in that, you know, in other words, it's sort of more like space-time than it is three-dimensional space. You can think of depth as being the analog of time in that it's ordered, rather than it being just three different dimensions. So mathematically, you can certainly fit spatio-temporal models to such data by treating depth as if it was time. Physically, I would always want to ask, is that actually a reasonable thing to do in context, or is there some better way to formalize the dependence in the depth dimension in a way that's qualitatively different to the dependence in the spatial dimension across the surface?
Thanks, Peter. Well, during our talk, some questions popped here about computational tools for doing that, and you already mentioned towards the end that PrevMap, specifically for the prevalence maps, are the key tool. I wonder whether even the maps you show are also produced within the R environment, or do you combine it with GIS resources to
No. These are all mapped within R, and the book with Emanuele Giorgi has an appendix on the way we do that. We use R packages which other people have written, which essentially are GIS-like, but it's not a full-blown GIS. So everything that you see, including the maps, is created within R, but using several packages written by people who are doing GIS-like work. Obviously, the alternative, and it's a matter of personal taste, is to download the output in plain files and transport them to a GIS if that's your preference.
So if I can just emphasize that this book with Emanuele Giorgi is a key reference for your talk, and it's a book released last year, right?
Yeah, correct. Yeah.
And
all of the code used for the examples in the book, including the mapping code, is actually on the web.
Yeah. So it's in the list of reference you provide in the beginning.
Yeah, sure.
Right.
If people do want to follow that, suggest they go to my website, because ironically there's since the book was published, the actual web address has been changed, not by us but by Google. So our websites will point people to the correct website, whereas the book web reference is now incorrect. But it's all there on our website. If people are looking for it and they can't find it, just email me.
Good. And I have a more general question here, Peter, about sampling size for spatial analysis. It's you mentioned a little bit of this when you were borrowing strength in the ROC discussion from different sources of information. But, well, I wonder whether you want to make any comments about restrictions of sample size to do some spatial analysis.
Yeah, it's always a question, and the honest answer is always that there is no magic number that says this sample is big enough and that sample isn't big enough. You have to do the design calculation and decide whether it's big enough for the precision that you are aspiring to. Now, however, I would say, as a rule of thumb, I would be very reluctant to try to build a geospatial model from less than 50 locations. Mm-hmm. However, if I've already got a geospatial model, in other words, if I've actually got some historical data from which I've built a geospatial model, and I want to apply it to some new data without having to re-estimate anything, I'm perfectly happy to analyze smaller data sets, because I do it in the knowledge that my predictive intervals will be wide. And as long as I refuse to let people use point predictions, as long as they're the truth, I'm comfortable, because what I'll get with my probability exceedance maps, the answer will be very simple. You can't be certain of anything. So it's not worth even trying to build a model, in my humble opinion, with less than about 50 data locations. Provided you express your results in terms of predictive probabilities, then they're honest, whatever the sample size is, and you can discuss with the owner of the problem whether the answer is precise enough for your needs. So, I mean, the Ghana example is a good example. It was not considered politically acceptable. This is the probability that each district has achieved elimination as a public health problem before doing any treatment. Now, because at the time Ghana was considered to be endemic for lymphatic filariasis, it would not have been politically acceptable to not bother sampling these districts to check for elimination. But my honest opinion is that if resources are scarce, you should take a smaller sample here than in some of these other areas where it's really not so clear, and you want to therefore get more information. So a good design for a follow-up survey might well focus on areas of maximal uncertainty rather than aiming to be, if you like, a level playing field, giving everybody the same number of samples. But I already know that the kind of design that will be acceptable, some of the work we've been doing is going to be implemented in the field in 2021 in Kenya. We've been specifically recommending on designs there, and they've already told me that the politically acceptable design will be to say, in each district, sample so many schools. It would not be acceptable to say, don't bother sampling here and sample 10 schools there. This would be considered unfair. And I have a lot of sympathy with that view. So you do need to temper statistical theory with a whole set of issues that are not reducible to mathematical calculations. But it is certainly true that the most efficient design will depend on not just what the true state of nature is, but what the specific question is. So if, for example, the question had been, what's the probability that ICT prevalence is less than 5%, then I would have absolutely said, don't waste your time sampling large swathes of the country. I can bet you my life it's less than 5%. As you get down to these very low levels, you're asking people to take a lot of faith in the model to really take something based on historic data only as being a certain answer. You really want some ground truth data to verify it. Sorry, that was a long answer to a simple question, but the simple answer is 50.
That's perfect. Good. A colleague of ours works in the field of hydrology, where he says that there is a lot of variation. Typically, it's a very erratic sort of thing. So he was asking whether we could have some formal mechanism to borrow information from regions which are more kind of stable to regions which are more like unstable or more difficult to estimate.
Yeah, if I've interpreted that question correctly, this is often, in my experience, a situation where there is a huge amount of value in thinking carefully about substantive covariates. And since the question was asked in terms of hydrology, I'll couch my answer in terms of another study we've been advising on, which is looking at schistosomiasis prevalence and monitoring for control of schistosomiasis as a public health problem. Now, schistosomiasis is spread by contact with a water snail. And so it is very strongly associated with water bodies. And what that means is that if you simply treat your sampling as if it's a level playing field, you really are going to miss the point completely. You have to sample very close to water bodies, and you have to sample in transects going orthogonal to the water body to catch that gradient because it's sharp. Now, there's no free lunch, and if you can't characterize that gradient accurately by, let's say, a regression relationship on distance from the nearest water body, then you are going to have trouble finding discontinuities. So discontinuities or sharp gradients are obviously much harder to find than shallow gradients, but at least if you are in a situation where you believe that that's the case, then that will certainly influence the kind of design you will use in the field. For example, including in your design some transects of closely spaced points, as well as some more spaced out points. And it will also mean that if you don't have covariates, you shouldn't be disappointed if you don't find very much spatial correlation. So again, subject matter knowledge and constructing covariates that actually have scientific interpretation is at least as important as subsequently fitting the stochastic model. I mean, for example, another of our examples, for example, is actually in a favela in Salvador, where, as your audience will be well aware, if you're walking through a favela, the environment changes very quickly and often very suddenly and discontinuously, and you have to capture that in fine-scale covariates. You can't do it just by a spatial correlation function.
Peter, this sort of connects a little bit with a comment by our colleague Max, which is related to the spatial domain, because what you see is pretty much related to the window you are looking at. He's saying that he works in marine ecology, I believe, with some concerns about pollution in marine environments. But his question was related to that, the extension of the study area and how it would affect the models and the choices being made.
Yeah, well, it's a question really of being realistic, I guess, is that there are two extremes going on here. One is you self-evidently cannot find out anything about small-scale spatial correlation unless you take data locations close together. But equally, you can't find out anything about large-scale spatial variation unless you span a large scale of observation window. So you need to balance those two things within your resource restrictions. Now, I don't think the underlying principles change at all in terms of the geographical size of the spatial region you're looking at. I mean, the same principles apply whether you're dealing microscopically or globally. But of course, in practice, the problems tend to be very different. And again, I wouldn't presume to analyze any marine data without a marine ecologist being involved in the project, because it's something about which I know nothing. I mean, over the years, I've learned something about tropical disease, but I still want to work with tropical disease experts, not on my own as a statistician. So that's the best I can say, really.
Great, Peter. Yeah, well, he always sent interesting questions. He was asking whether statisticians would save the world on that, and basically what you said, you help people who are concerned about these problems. It's not our duty.
Yeah. You may have heard me say before, Paolo, and I'll say it to all your colleagues, statistics is fundamentally a benevolent parasite. It only adds value to other disciplines. It's of no value on its own, but it is benevolent. It does add value, and often substantial value.
And with this comment, I think we finish the questions here, Peter. But I'd like to point two things. First, it's nice to see the triangle, the science triangle you brought to us, because we use your book as a reference for the first lecture, and we pointed to that triangle. So it's nice connection, see the triangle there. And second, your example, especially the one with pre and post intervention, has a clear connection with our next lecture on assessment of public policies, which I think brings a nice bridge. So with this, I think we can stop here. I would like to ask you to stay with us a little bit after we close for interval. But while our students are here, I'd like to thank you so much for this beautiful lecture. It was a great pleasure to hear you again. And the course and our university, thank you very much for highlighting, to provide these lectures for us on our student data and spatial analysis, which certainly is a big asset for our course. Thank you so much.